Okay, so chapter 11, it's introduction to the nervous system and nervous tissue. So something that we are going to see in this chapter is the classification of the nervous system, and we're going to see what are the functions of these different structures within this classification, including PNS, CNS. We're going to differentiate what is the difference between afferent division, efferent division, and their subclassification. And we're going to compare the functional branches of the nervous system, which are somatic and autonomic. We are also going to see the parts of a neuron, classification of the neurons, the function of the different parts of the neurons, and supporting cells, including their names, what do they do, where they are located, all of these different supporting cells. And we're going to revisit again the physiology of excitable cells. So basically, we're going to look again at what is resting member potential, what is action potential, and their phases, and what is the importance of having these periods within the action potential, and how you transmit these action potentials along these membranes. Also, we're going to see the difference between electrical and chemical synapses, and we're going to see different types of neurotransmitters, what are the different types of action potentials in terms of once you move it from one cell to the other. So we're going to see all the details of this, and we're going to go into neural circuits and classify them as well according how they carry the information. So now, first, what is the nervous system and why is essential? So the nervous system, it is these structures that we have within the body that are in charge of processing information and then responding to this information. So we have sensory information all around the environment and also internally, and then we have to process this information, what it means, what do we need to do, how do we react. So that's what our nervous system do, and also we control the function of most of the system of our body with this nervous system, and we maintain homeostasis through this. So then we process sensory input, we integrate this information, basically we know what we are dealing with, and then we respond. So we generate an output. It can be motor, some cases, not all the time, but that's what we do. And then our nervous system is subdivided into two major branches, CNS, or central nervous system, and PNS, or peripheral nervous system. CNS is made out of two parts, brain and a spinal cord. And everything that is outside the brain and the spinal cord will be peripheral nervous system, or PNS. So CNS is protected by the bones of the skull and also the vertebrae. And within a way, both are connected, CNS and PNS, of course, through these nerves that coming in and out of the spinal cord and the brain. So, major functions of the nervous system: sensory function, detect internal and external stimuli. This morning when you woke up, maybe you were feeling hungry, and then you went and eat something. Well, that hungry sensation was your sensory input. And then that behavior of going and eat something to not feel hungry anymore was part of the integration of the nervous system. And that behavior change, well, it was to satisfy your body. So we detect external stimuli. You're listening to my voice. That's sensory information that is going into your brain. You're feeling the temperature of the room, if it's comfortable or not. Maybe we will hear an alarm going off from the fire system. And if it goes off, we already know what to do because we were trained to respond. Maybe we have to exit the building because of that. More or less, I don't smell so much about someone wearing a perfume right now. But that's sensory information going into your body as well. What if something is burning? That's sensory information. Whatever you're seeing right now, that's sensory information. And then integration or integrative function will be processing, interpreting sensory information to make decisions. Well, you also sense internal stimuli. That internal stimuli can be how full is your urinary bladder. If your urinary bladder is empty, well, you don't feel the urge to go to the restroom. But as it's getting full, and you're sitting here for almost 2 hours, you will be like, well, I can hold it. I can hold it. And then Sully's going to say something important, and you're like there, like. But then, if the time comes where you cannot hold as much urine in your urinary bladder, then no matter what I say, or as important as it is, you just step out, go to the restroom, and then come back. So that's interpretation of internal and external stimuli. What if it is getting hot? Well, what would you do? Well, you remove layers of clothing. So that's basically how we function. So motor function is sending commands to muscles and glands to respond appropriately. Well, if it's too hot, then not only you will use your muscles to remove your clothing, but also your sweat glands start to release sweat so that you can drop the body temperature. That is through motor output. So you have nerves that goes into your glands or goes into your muscles, and you have a motor function. So here we have an example of how we use the nervous system. So in this case, we have someone playing soccer, and then the person is looking at the ball as it's reaching towards this area. By gravity, it's going to go into this part of the floor, but before it hits the ground, the person knows which muscle to use. Maybe if it's this side where you don't have players, and you are like if you were the audience of that soccer game. Of course, there are no players there. You are the audience only. So you're the sports fan. It doesn't make sense for me if the ball is coming this direction to hit it with my with my left foot, right? Why? Because, oh yeah, then I pass it to you and then you start playing. No, you can't. So I have to visualize where the ball is coming, which foot am I going to use, and then lift my upper limb, and then not use every part of the foot, just the part that is right to direct the ball to where I want it, which in this case, it will be the middle of the field. So it will be the right foot. So then you have a visual input going into your eyes. And then from there, you send a command into the premotor cortex and then to the motor cortex where we have nerves that are going to go and travel through the spinal cord and connect to make a synapse or a relay to a motor nerve that is going to contract these muscles so that you can flex your knee and then dorsally extend your leg, and then you can hit the ball. So that's part of a reflex. So you have sensory input here that is going to sensory receptors that detects the stimuli, and then you send that signal into a particular area of the brain to process that information, and then you make sense of that information, that's integration, and then you have a motor output. And then you perform that response. Okay, so that's the general function. Now, so we said that the CNS, or central nervous system, is made by the brain and spinal cord, and the PNS by the cranial nerves, spinal nerves, and peripheral ganglia. Outside of the spinal cord, we have little structures that are almost round that are called ganglia. That's where we have the body of the neurons that connects to axons, and then you have certain nerves either sending information or receiving information. So how many cranial nerves do we have? 12 pairs. And usually you abbreviate cranial nerves as CN. And then we have 31 pairs of spinal nerves, or SN. Now, for spinal nerves, this is the subdivision by region. We have 8 cervical nerves. We have 12 thoracic nerves. We have 5 lumbar nerves, 5 sacral, and 1 coccygeal. 8 plus 12, 20, plus 5, 25, plus 5, 30, plus 1, 31. So we have 31 total spinal nerves. For the cranial nerves, most of them you already know the names, like you remember trigeminal, you remember olfactory. We will see that again, but they have specific names, and they have that name and that number. Now, for the spinal nerves, all you need to do is tell the name of the region plus the number of the nerve. So the first cervical, second cervical, third cervical, 12 thoracic, 5th lumbar, etc. That's how you call these spinal nerves. Now, how many cervical vertebrae do we have? How many vertebrae do we have in the neck? 7. But we have 8 spinal nerves. And the reason for this is because each spinal nerve exits above the edge of their superior vertebrae. So what it means is that C1, or the first cervical nerve, exits above the upper edge of the atlas. And then C2 above the upper edge of the axis, or below the atlas, and like that. So then the 8th cervical nerve is located between the thoracic, the first thoracic, and the last cervical. So, and then the rest will be T1 through T12, and so on. So here is this anatomical classification of the nervous system. So what does the brain and the spinal cord do, or makes? The CNS or the PNS? CNS. So this is the CNS. And then all of these nerves will be PNS, peripheral nervous system. So these spinal nerves exit directly from the spinal cord, and the cranial nerves from the brain, but some of these like the lower cervical and the upper thoracic, they form branches or networks that we call the plexuses. So we have a cervical plexus, we have a brachial plexus, which is this, and then we have a lumbar plexus, which is this, and a sacral plexus, that these two together form that lumbosacral plexus. And they control specific areas of the body, like the cervical, the neck, the brachial, the arm, and the upper limb. And then the lumbosacral controls pelvic area and the lower limb. So again, they merge, they form networks. So these are the networks. And then from there we have the functioning. So now let's look at the subdivisions of the PNS, or peripheral nervous system. The PNS is divided into sensory or afferent and into motor or efferent. Sensory, again, receives sensory information from touch, temperature, pain. And the sensory is subdivided into somatic and visceral. Somatic will be that sensory information coming from the skin, muscles, and joints. If you ever had a spider or a bug crawling through your skin, it feels ugly, especially if you are unaware. That information is carried by the somatic, because it's coming from the skin, and it's sensory. It's part of the afferent division of the PNS. Visceral sensory conveys information from internal organs. We are not necessarily aware of this information all the time, unless it's something like, for instance, when our urinary bladder becomes full. But what type of sensory information can come from internal organs into the nervous system? Well, what is your heart rate? What is your blood pressure? What is your distension of internal organs, of the stomach, the large intestine, small intestine? So we have stretch receptors that send this information into the nervous system. So that's visceral. So visceral refers to internal organs. Now, motor or efferent division of the PNS. So we have somatic motor. Somatic motor controls the muscles that are attached to the skeleton. And then we have autonomic nervous system, or ANS, that has two sub-branches, the parasympathetic and the sympathetic. So the ANS controls involuntary functions of the smooth muscle, cardiac muscle, and the glands. So the heart contracts more or less like around 70 beats per minute on average, 80, because we have this ANS making the heart contract. When we eat the food and goes into the stomach, from the stomach goes into distal portions of the digestive tract, because we have muscles that contract and relax under the influence of this ANS, the vagus nerve specifically for the parasympathetic system. So what is the difference between somatic and autonomic motor divisions? Well, somatic is voluntary, so we can control the contraction of our skeletal muscles. And then autonomic nervous system is involuntary. So it controls functions like contraction of our heart, so it gives us particular heart rate, also help us to breathe and to digest. No one in here can tell to the heart, well, beat this many times per minute. So ANS is involuntary, while somatic is voluntary. So here is the structural and functional division of the nervous system. CNS, brain and spinal cord. And then PNS includes the cranial and spinal nerves that links all the structures of the body to the central nervous system. That's why CNS basically controls many of these functions. So sensory comes from joints, skeletal muscle, skin, and this is somatic. Like if you have pain in your knee, you may have fall down or made a bad movement, that sensory information goes into the CNS. If you have an irritation of your skin, that information goes into the CNS. Urinary bladder full or empty, that information goes into the CNS. That's the visceral sensory division. If you're in an empty stomach right now and feel hungry, that visceral information goes into your CNS. And then you will have a motor or an efferent division that controls your skeletal muscles. And then a visceral division that it is controlled by the ANS that makes your heart to contract, your smooth muscle to contract, and your glands as well. Okay, so the neurons. Neurons have different parts, and they will be named based on the terminology that it is used for nervous system. Just like as for the muscle, we have in the muscle fibers or the cells of the muscle, same organelles but with different name. Similarly here with the neurons. So we have the bulk part of a neuron that is called the body or soma. In this body or soma, that's where we have the nucleus and the organelles, most of the organelles of the neurons. One of the organelles that is very abundant in the neurons is the nasal bodies, which are the ribosomes. The neurons produces a lot of neurotransmitters that many times are peptide based, so that's why we have these nasal bodies. So what is the function of the body or the soma? Well, to receive and process information. And this body or soma has extensions that are called dendrites, like that they can be compared like branches of a tree. And these dendrites receives information from other cells. And then at the end of the dendrites, they have a little extension that is called telodendria. That is the one that specifically is connected by other cells. And it won't be a physical connection. There is a space between the cells that connects the neurons that is called the synaptic cleft or gap. But there is an electrical chemical connection because one cell will release chemicals so that the other cells will catch those chemicals and then convert it into an action potential. Let me give you an example. And Lucas is not here, but ready?
Oh, I'm back again.
If you receive blue color, that means standing up, black color, stand down, okay? So I'm going to send it, you will catch it. There is no physical connection, but this is the neurotransmitter. You have this, which is a ligand, and your hands will be the receptor. And then depending on what neurotransmitter it is, this one is going to be acetylcholine. It will open certain channels so that you depolarize and perform an action. This one will be GABA, maybe. It will, or any other neurotransmitter. So it will open chloride channels so that you don't depolarize your cell, you hyperpolarize the cell, and the cell will stay in resting membrane potential, okay? So, I'm going to try it at the same time, so be ready. 1, 2, 3. Okay, so depolarization, hyperpolarization. Chloride channels open, sodium channels open. Sodium channels, sodium is positively charged, and then chloride is negatively charged. So this makes the cell more negative, this makes the cell positive. So that's a chemical signal that it was transformed into an electrical signal. I will drop it. Pretty bad catchy. So then this space in between us was the synaptic cleft, but there was still communication. So that's how our nerve cells work. So the plasma membrane that covers the neuron is called neurulema. The skin that was covering the hands that were catching these markers is the neurulema. Okay? And then the axon hillock is the connection between the body or soma and the long extension from the neuron that's called the axon. So it's like a little neck between the soma and the axon hillock. And this is where you summate or add the stimuli for the action potential to happen. In this case, I just released one neurotransmitter. But what about having a code where you say, I have to have this specific amount of neurotransmitters for this action potential to happen? It's like if you go to purchase an item in a store, or if you go to a restaurant, right? And then you're trying to get a dish that costs like $20, and you're trying to, you say to the server, well, I have only 10. It would say, well, you cannot choose this part of the menu, you can choose this. So you have to have a specific amount of neurotransmitters sometimes to produce an action potential. So we will see that later. And then the axon is the longest extension of the body, and it transmits the impulses away from the cell body. And then the plasma membrane that covers the axon or surrounds the axon is the axolemma. And the cytoplasm within the axon is called the axoplasm. And this one allows the transport of molecules from the body into the axon terminal, which will be the ending of the axon. And the axon terminal sometimes it has like this little expansion that looks like a button. So it's like a button-like extension. And function of the axon terminal to store and release neurotransmitters. So here are the parts of the neuron. And this neuron is classified as multipolar because it has multiple extensions. So this is the bulk of the neuron. What is the name of this? Soma. And then out of the soma, we have these branches looking like if they were branches of the tree. What is the name of these branches? Dendrites. And it's not labeled in here, but the tip of the dendrites will be the telodendrite. And this dendrite receives information from the cells, the other cells, and then bring it in here into the body. So that the body with the nucleus and all the ribosomes and the organelles will make sense of what it needs to do, integrates the information, and then that information, if it needs to be converted into an action potential, then you convert it in this area that is like a little neck. So this will be the axon hillock. And then this long extension will be the axon. And then the end of the axons will be the telodendrite. Sorry, the synaptic terminals, the axon terminals. Now, it's not labeled in here, or well, yeah, it is here, but all this axon is covered by the plasma membrane that we know as axolemma. This is an important point for, in general, for the lab guys or for wherever you are looking at some items in diagrams or figures, pictures. Sometimes when they're trying to tell you certain parts, well, they have to label in a particular area. They cannot be labeling everywhere. But what it means that they are labeling here axolemma next to this axon hillock or next to the axoplasm. What it means is like the entire membrane that covers the axon is called axolemma. It's just that it would be impossible for labeling everything, but it is understandable then that as long as the membrane is covering the axon, it will be called axolemma. And axoplasm, of course, not only existing here, it exists everywhere within the axon. It is just the cytoplasm of the axon. Okay, so just make sure that you know that. And of course, this is a synapse between a target cell and an axon or a neuron, so that these target cells perform a function. And then, of course, here's the nucleus. These organelles will be the nisul bodies or ribosomes and the ER. This is the mitochondria, and this is the smooth endoplasmic reticulum, and these are neurofibrils of actin and myosin that maintains this shape of the soma. Any questions of this neuron? Okay. So, functional regions of a neuron, receptive. Just like our eyes receive sensory information, what we see, and our ears, what we hear, this is our receptive field. Well, this is the receptive field of a neuron, and this is the conducting field or region. So action potentials starting here and then travels through the axon. And this is the secretory region. That's where we send the neurotransmitters. Any questions of this part? So neurons, how they are classified based on a structure. Well, if the neuron has one axon and multiple dendrites, This will be multipolar. Where do we have them? In the spinal cord, like the motor neurons in the spinal cord. In the brain, in the cerebral cortex, we have the pyramidal cells, and also in the cerebellum, the Purkinje cells. So we have these neurons where we have to receive information from many places. So they are located mostly in the CNS or the motor pathways of the PNS. Bipolar neurons have two extensions, one that is an axon and the other one that is a dendrite, that are of course connected to a body. And they're found in sensory organs like the retina or the olfactory cells, olfactory epithelium. And then we have some neurons that looks like they have only one extension, that are called pseudo-unipolar. Pseudo, this part means false. Now, this extension that these neurons have, it's a short process that splits into two axons. And it doesn't have true dendrites. Those are the pseudo-unipolar. So we have it in sensory neurons that help us to feel sensations like touch, pressure, pain, and vibration. So we have it in the ganglia, in the PNS, this pseudo-unipolar. So that was the classification based and structure. Now the classification based on function. So if your neurons connect to the two different neurons or bridge information between them, this we call interneurons or association neurons. These typically are structurally bipolar. And they are located only within the CNS, connecting motor and sensory neurons. They process information and then they integrate and interpret sensory input to formulate a response, like in a reflex arch. Motor neurons, it's the other classification. These are also known as efferent. These ones basically starts in the CNS, like in the anterior part of the spinal cord, and then they connect into peripheral effectors like muscles or glands. An example of these will be multipolar neurons in the brain. And then we have sensory or afferent neurons that receive information from peripheral portions of our body, like the skin, muscles, and joints, and then they send that information into the CNS. They're afferent. Another way to remember is arriving. And these connect into the posterior part of the spinal cord. And these type of neurons can be bipolar or pseudo-unipolar. They're located in the peripheral ganglia. And they are also located in the retina, in our part that is behind our eyes, and also in our nose, in the roof of our nose. That's where we have sensory neuron for olfaction. So they are located in the olfactory nerve. So here is the different types of neurons that we have. So this will be motor, efferent neurons, and interneurons. While these ones will be sensory or afferent. Afferent or arriving, they connect into the posterior spinal cord, some of them, and these ones are efferent or exiting. They are connected into the anterior spinal cord, some of them. So this one, it's a multipolar. It has several dendrites and it has this axon. So this is a spinal motor neuron, for instance. This one is a pyramidal cell that has these dendrites and this axon. This is located in the cerebral cortex. So these are the ones that we use to learn. And then this, it's a multipolar neuron that has these multiple dendrites and this axon. This one is called Purkinje cell. This one is located in the cerebellum. It's a tiny organ that we have here behind our brain and inferiorly that help us to maintain equilibrium so that we don't fall. And also help us to have fine movements because it provides a circuit between the cerebral cortex and the cerebellum that instead of making a gross movement, like for instance, if I want to get that marker, I don't do this. This is a gross movement, and I don't miss it. With that cerebellum, you produce a fine movement and you know exactly where to get it from. When people have damage to that cerebellum, to these cells, you ask them, extend your arms, and then extend your index finger. And then close your eyes or have your eyes open, touch the tip of your nose bilaterally. Anyone who doesn't have problems with the cerebellum is supposed to do this. Well, people who has problems with the cerebellum, even with eyes open, they start doing this. They have intention tremor. And they start doing this until they get closer, and they hit it. They hit the right area. And they can walk like if they were drunk because they don't coordinate well their movements. So those are the Purkinje fibers. These ones can receive connections out up to 200,000 cells. That's how they are so important. And then this sensory or afferent, this is a bipolar neuron. It has a single dendrite and a single axon. We have it in special senses, like in the retina, so we can see. Also in the olfactory epithelium, in the roof of our nose, so that we can smell. And then this is a general sensory neuron. So it's pseudo-unipolar. It has a peripheral axon and a central axon, this single extension. That's why it's considered pseudo-unipolar. And then it has receptive endings in here and secretory endings in here. So these ones are located, for instance, in the dorsal ganglion of the spinal cord. So we look at the information about neurons, the main cell, the structural and functional unit of the nervous system. Now, our nervous system also have supporting cells. These supporting cells as a whole, they are called neuroglia. And we have different types of neuroglia. And we have neuroglia in the central nervous system, and we also have neuroglia in the peripheral nervous system. Because in both PNS and CNS, we have neurons that need support. So the CNS neuroglia will be astrocytes, oligodendrocytes, microglia, and ependymal cells. And the neuroglia from the PNS will be the Schwann cells and satellite cells. Functions of the astrocytes. support neurons. Basically, if a neuron needs calcium, it grabs calcium from the bloodstream, put it next to the neuron. If the neurons have excess of potassium, it removes it and then put it into the nervous system, into the circulatory system. So it gives chemical support, so it maintains particular levels of ions that the neurons need, but also provides an structural support because the astrocytes have extensions that holds the axons so that the neurons won't be floating. And part of these extensions is wrapping around blood vessels like the capillaries and forming the BBB, blood-brain barrier. With this blood-brain barrier, we prevent molecules that can damage our brain to come into the brain. But that's not everything. I mean, if this blood-brain barrier will be foolproof, then many people won't get infections in the CNS or get damage into the neurons due to the use of drugs or alcohol. I mean, this barrier is not completely impermeable, but still it is impermeable enough that protects the neurons for most of the damage that it can happen without it, okay? Now, oligodendrocytes are cells that produces a substance that is called myelin. Myelin is like a lipid base with some proteins, molecules, that coats the axons. And with this coating around the axons, the electrical signal can jump between spaces in myelin. These spaces in myelin are called the nodes of Ranvier. And that makes the action potential to travel fast. Sometimes you want the action potential to travel fast because you are transmitting important information. Like what type of information? Well, injurious sensory information like pain. So if you have had, or if you have in your home, these cast iron pans, they can get really hot. And then if someone doesn't warn you that this cast iron was used before, a few minutes before, and the stove is off, and then you by accident thinking that it's cool, you grab the handle, feels so bad. I mean, you will burn your skin. That information, of course, needs to go quickly into your brain so that before you think that it's painful, you withdraw your hand. Like this, it's a reflex. You contract your muscles and then you withdraw your hand. So these neurons that brings that information has to be myelinated so that the action potential travels fast and then you receive that information quicker into your brain. By the time that you feel the sensation of pain, you are aware of that, you already have withdrawn your hand and you don't damage your muscles and your skin with that handle. So insulation of neurons can be, by myelin, can be considered just like as a copper wire, that it is insulated, so that the electrons that are flowing through this copper wire don't go externally and someone touches that and the person can get even electrocuted. Okay, so that's myelin. And it's produced by oligodendrocytes in the CNS and by the Schwann cells in the PNS. Now we have macrophages. Macrophages are cells that can eat debris, it can eat bacteria or dead cells. So the macrophages of our CNS are called microglia or microglia. So they are part of the immune system of the CNS. And then we have surrounding the cavities in our brain that we call ventricles, we have ependymal cells. These ependymal cells produce and circulate a fluid that maintains our neurons moisturized, that we call the CSF, cerebrospinal fluid. And then we have cells in the PNS that surrounds axons and the cell bodies that are called satellite cells. Any questions about these neuroglial cells? So they're not the ones that process that information or receive that information, but they are the ones that supports the neurons. It's like parents. They're not learning for you, but they support your learning. So here we have all these different types of cells then. So we have the main players of the nervous system, which will be the neurons, which are these. And then we have the astrocytes. The astrocytes extend their extensions, and they physically connect the neurons and also the blood vessels, forming the BBB, blood-brain barrier. So these extensions are like little feet, and these are called end feet. And then here we have either Schwann cells or oligodendrocytes. Oligodendrocytes myelinate multiple neurons, while the Schwann cells only one single neuron at a time. And these are microglia. These are the ones that eat debris, eats bacteria. And then here we have these ependymal cells. These are the ones that line the ventricles, the cavities in our brain, and produce and circulates the cerebrospinal fluid. And these ependymal cells, we also have them in the central canal in the spinal cord. So this is myelin. And these gaps expose the axolemma so that the action potential can migrate fast. And this is the actual microscopy, light microscopy of the neurons. You have seen this before in the histology chapter. So this is the body of the neuron, this is the nucleus, and this will be either dendrites or axons. How do you know? Well, based on length. The longest is not seen in here, but I imagine that they have seen the entire length whenever they took this picture. The longest will be the axon, the shortest will be the dendrites, and these tiny cells in here will be the neuroglia. Neuroglia outnumber the neurons, but they don't have as much function. So here we have PNS sample from the ganglion, dorsal root ganglion, specifically that's how it's called. This is the spinal cord, this is the spinal nerve. So this is the sensory root, this is the motor root. So if you magnify this, you will be able to see these peripheral cells surrounding the body of this neuron, this sensory neuron. And here we have a central axon, this is a peripheral axon. So this is a pseudo-unipolar cell, because this looks like if it was one extension when it's actually two. So here will be central axon, and here will be peripheral axon. And then here's the body with these satellite cells. And what myelinates? So this is myelin. These cells, the Schwann cells, or neuroleumocytes. So here's the myelinization process of these neurons. So these are segments of myelin. If you make a section of this, you see it, this is the axon, this is the myelin, in a scanning electron micrograph. So everything starts here at the central part surrounding the axon. So this myelin formation will be going around until you form a thick layer of myelin. And then if it's a peripheral nervous system cell, will be myelinated by a single Schwann cell. If it's oligodendrocytes, they can myelinate several axons at the same time. This is another example of unmyelinated axons and myelinated axons. So in this case, we have unmyelinated axons in here. And then here we have one of these cells of the peripheral nervous system cell, which will be a neuroleumocyte. But in this case, it's not myelinating these axons. So what happens when we injure one of these cells? So this is the body, this is the axon, and this is the myelin. If you sever one of these axons, there is a missing connection between this proximal part of the axon and this. So this one will disintegrate, and this target cell, in this case an skeletal muscle cell, will start becoming atrophic because it's not going to receive acetylcholine. Eventually, with time, these will reconnect. So first you miss this part, and this one will, this part will regrow with the aid of some peripheral cells or satellite cells. And then once you have growth processes going on, they would follow a track, and then they will reconnect to this muscle cell, and the muscle cell will gain back its thickness. Now, if this nerve will be severed close to the body, then this reconnection doesn't happen. Peripheral nerves, usually they reconnect and they have a function again, but sometimes if it's so severe damage, that won't happen. For sure, that regeneration that you saw before, it doesn't happen in the CNS neurons. That's why they are the most important. And whether if it's a chemical injury or a physical injury, it's very important to protect so much your spinal cord and your brain. But that doesn't mean that if you have a damaged nerve in the peripheral nervous system, you can recover. Sometimes people end up with numbness in certain areas of the body because the nerves, they don't reconnect or reconnect. Okay, so that was the gross anatomy and the function of all these cells, supporting cells, and the neurons. Now let's look into action potential again. So we have gradients of ions, and this gradient of ions is regulated by the transfer of ions in and out of the cells through voltage-gated channels. And these voltage-gated channels, they have a specificity. So we have calcium channels, we have sodium channels, and we have potassium channels, chloride channels. So only those ions can cross through these specific channels. Right now, all of you, at least in here at this time, you are A&P students. So only A&P students pass through this door. So that will be the A&P channel, okay? So now, these channels can open and close in response to changes in the electrical potential or the voltage. That's why they are called voltage gated. And then the resting member potential of the neurons can vary from minus 70 to minus 90 millivolts. Some neurons are more negative, they have minus 90, and it will take more depolarization or more ions to reach a threshold and then to start an action potential. Now, this voltage of resting member potential is maintained by this pump, the sodium-potassium pump, and potassium leaky channels. Leaky channels are always open. And voltage gated, they have a gate that can open and close. Now, anesthetics, local anesthetics, can make this sodium-potassium pump to not function temporarily, and that prevents the exchange of ions between this compartment, the intracellular and extracellular compartment, and that is what can lead to precisely not having an action potential that can lead to the sensation of pain. So anesthetics then block these channels. So here is activation and deactivation of voltage-gated ion channels. So this is resting state above and activated state below. So here we have this activation gate closed and then the activation gate open. When it's closed, there is no flow of electrons, ions, between the extracellular fluid and the cytosol. The cytosol here is shown in yellow color, extracellular fluid with blue color. So when you have voltage applied, this will open and then potassium escape. Why? Because you have more potassium here in the cytosol than in the extracellular fluid. What is the voltage for you right now? The voltage for you is going to be 145. You are all potassium. When 145 arrives, you will all exit. So that door will be open. It's still open, but I can close it and you open it and you exit. So that's your activation gate. Now, for potassium is just active or inactive. Now for sodium, we have this activation gate that is closed and this inactivation gate that is open. So it has three parts. And this is in resting state. In resting state, this inactivation gate is open and the activation gate is closed. Now, this side is going to be the right side activated state and the left side is going to be inactivated state. So then what happens when you have a change in voltage? So these activation gates open and this inactivation gate will be open. So then that is going to make sodium to rush from the extracellular fluid to the cytosol. Why? Because you have more sodium out than in. We had to wait a little bit outside because there were some people in before 12:30. So we were accumulated out. As soon as these two students come out, then we all, since we were actively concentrated there, then we move in here. So that's when the the activation gate opened. And then once you have enough sodium in here and you don't want to bring more sodium, what you will do, this inactivation gate will close here and then no more sodium can come in. That's the inactive state. Any questions? So that's that or this for you. Okay, so we will end here. If you don't have any questions, I will see you